All right. Thank you once again. Uh, is my screen visible?
Yes, sir. Yes, sir. Your screen is
All
visible.
right. Thank you so much. Sorry for that little break in transmission, and I'm very glad and opportune to be here today. So, just like you've heard, my name is Ian Osoro. Currently, I'm a PhD student. I study at SRM in India. I'm in my third year, and basically, what I'm doing for my PhD research work is on clinical trial in neurology. So that's where I'm majoring in. I did my bachelor's in pharmacy, and then I did my master's in pharmacy practice, and then now I'm doing my PhD in pharmacy. So thank you so much once again for the founder PASH, and then for the esteemed leadership and for all members present. I do not take it for granted. Thank you so much for granting me this opportunity here to just share a few of the things that I've learned. And please feel free to interact at the end of the session. And I would just also want to mention that it's a topic that I chose out of the desire to share because I feel like as Africans, we are kind of left behind sometimes when it comes to things like artificial intelligence or in innovation. So by the time we catch up, we are usually a bit very late. So I think this might steer any one of us here that plans to go into the research field in regards to healthcare research or clinical trials and so forth. So thank you so much. And yeah, I think our moderator said that I am a guest. So I think I'm a member here. I'm at home. So please, yeah, I feel very at home and I feel very happy. So let me just begin my presentation without much wasting time. So basically, today I'll be talking on artificial intelligence in clinical trial. So I'll just be defining everything, and then step by step we'll want to understand what does it mean to go from uh, sorry. All right. So the next slide, we'll be talking what are the contents for this topic that I've chosen for today. So I'll be talking on the introduction to clinical trials because it's possible some of us here may not really understand or know about a clinical trial. Then I'll talk on the phases and the challenges of clinical trials. Then I'll speak on the introduction and the evolution of artificial intelligence. I'll talk on the artificial intelligence technologies in clinical research. I'll talk on the AI applications in clinical trial design and recruitment. Then I'll speak on AI in data monitoring, analytics, and safety. And then finally, I'll speak on the regulatory ethical challenges and future directions as regards to AI in clinical trials. So basically, what is AI? I mean, what is clinical trials? So here we see that define clinical trials as systematic research studies which are conducted in human participants to do what? To evaluate the safety, efficacy, pharmacokinetics, the pharmacodynamics, and overall therapeutic value of medical interventions such as drugs and vaccines, biologics, medical devices, surgical procedures, or behavioral therapies. So we see that they are conducted according to internationally accepted ethical scientific standards such as Good Clinical Practice Declaration of Helsinki. So just to give a brief overview, a clinical trial is basically a is a research work that is done by a team, maybe a student or an organization, and then it is conducted on human participants. So we know that for those of us that in the pharmacy background, it takes about 10 to 15 years for a drug since its inception to the time the drug comes to marketing. Yeah, and that's a very long period of time. So we have preclinical development of drug, which is what I'll speak about in a very brief as we go on, and then we have the clinical part, which is also what I'll kind of major on. So a clinical trial, basically, just imagine that or think that you are a principal investigator. You want to test whether drug A has an efficacy in the patients. Yeah, you have proven it has some efficacy in silico studies. You have proven it has some efficacy in animal studies. Now you want to prove it in humans. So we want to see how that happens, and then how can we integrate artificial intelligence in helping these clinical trials? So we see, for those of us also that are not aware of the Declaration of Helsinki, this was as a result of some unethical kind of standards that were being practiced back then, like in the 1960s, 1940s. So this led to good clinical practice and Declaration of Helsinki, so that there will be ethical standards in conducting a clinical trial. So what are part of the Declaration of Helsinki? That there should be an informed consent. So a patient should not be brought into a clinical trial without being told what they are going to be doing. So you don't just blind them and then maybe give them money. No. So we see that these guidelines are also from organizations like the World Health Organization and also the US Food and Drug Administration. So what's the importance of clinical trials in drug development? So we see that clinical trials, they are essential because they determine whether a medical intervention is, one, safe for human use. A medical intervention may seem to be having potency among animal studies, what we call preclinical studies. Yeah, it may show some efficacy, but when you bring it to the human, because of the different dynamics and kinetics that humans have that are different compared to animals, then you see that that medicine or that drug, if it's not being efficacious. So we have to do a clinical trial before any medication comes into market to ensure that, one, it is safe, two, it is effective. So this effectiveness we assess by checking, okay, how much of the disease has it been able to stop? The disease progression, is it stopping the disease progression? Is it giving the patient a better quality of life? So these are some of the ways that we measure the effectiveness. We try to assess, okay, apart from the biomarker parameter, what other parameters uh, in a patient or in a clinical study are there to ensure that this medication or this drug is safe. So we also see that we can do a clinical trial to compare and to see whether it is better than an existing treatment or placebo. So basically, a placebo, we know it's what is a dummy drug. We call it a dummy drug. So when you have a clinical trial, especially a randomized double blind, then you have to do placebo. Placebo means you give the patient or the participant a drug, a form of a drug that there is no active pharmaceutical ingredient. Yeah. What makes a drug is what? As a pharmacist, we know that what makes a drug is what? Is the API, majorly, the active pharmaceutical ingredients, and then the excipients, which can be like the sweetening agents, coloring agents, and all those things. So for a placebo drug, you don't put any active ingredient, so it's just there to act as a dummy. So we want to check whether this drug that you are trying to test, is it better, yeah, or is it better than the existing treatments or than the placebo? Because the placebo effect is when a patient, in their mind, psychologically, they think, if I take this new drug, you tell them this is a new drug, although you have not put any active pharmaceutical ingredient, by themselves psychologically they believe they'll be healed, and then they start their body cells start regenerating. So that's why we also have to put placebo in clinical trials. So we also have to check whether it is suitable for regulatory approval. So this is the importance of having a clinical trial in drug development. And then quickly we go to the major roles in drug development. So we are going to be assessing what the evaluation of safety, because as I've spoken before, that one of the key things we assess when you're doing a clinical trial is what is the safety. Because before you bring your drug to the market, you can say this is the best drug ever, this drug does not have any ADR, blah blah blah, but we have to ensure, is it safe? Before any medicine is used in the public, is it safe, or is it going to be bringing chaos? Is it going to cause teratogenicity? Yeah, because we have seen like the thalidomide tragedy, which happened long back in 1960. So when they gave the pregnant women this drug, it ended up causing teratogenicity, whereby the young babies that were born, the infants, they were with deformed limbs. So we also have to ensure that, most importantly, is it safe? Then we have to assess the toxicity levels. How much toxic is the drug? And then we have to assess also what is the maximum tolerated dose. We have also to assess the long-term safety concern. Is this drug um, is it over time? If you use this drug for 6 months, if you use it for 1 year, if you use it for, let's say, when we have chronic conditions such as diabetes, hypertension, heart conditions, they are usually very prolonged conditions. Kidney issues. So when you're bringing a new intervention in this kind of patients, then we have to ensure that this medication can have a good long-term safety. Yeah, otherwise it will may after some time bring about some undesired outcomes. So in evaluating of safety, to ensure that unsafe compounds, they are eliminated from before the public use. So also the second thing we said is the assessment of efficacy, and then we see that here uh, we evaluate whether the investigation of product, which is our new proposed drug, biologic, or whatever we want to bring into the market, it's having the intended therapeutic effect. Let's say I'm proposing drug A can cure Alzheimer's disease, which is a neurological disorder. So once I say that drug A is able to cure, or this intervention that I'm bringing is able to cure, and as we know, there's no therapy ready, or there's no medication that in that sense as of now that can cure that, except the Kembe, which is just there to uh, stop the progression, but it doesn't cure. So when you bring such an idea, then we have to present and assess it by a clinical trial. Okay, we measure stuff like the biomarker, the psychopharmacological test, the mocha test. How is the patient are responding to the therapeutic effect of the drug that you have given? So we see here that the types of clinical trials by outcome focus, they are majorly three. We see that there are superiority trials, which they aim to demonstrate whether which one is better. Is drug A better than drug B? And then non-inferiority trials. So we know that, for example, that drug A is the standard drug. So we want to show that even drug B that effect, they produce same therapeutic outcomes in the patient. So this is what we do when assessing the efficacy. So this we can see on the slide I put there some of examples we can use to assess efficacies. You have to be very uh, direct. How do you want to assess efficacy? For example, in cancer, you have to check for, is there any tumor shrinkage? If you have, let's say, for any neurological disorder, is there an improvement in cognition? Whether it's in hypertension, is there a reduction in blood pressure? So forth and so on. We have to ensure that our efficacy parameters are well prepared before we go on for a clinical trial. So thirdly, we go for the dose optimization, and this is conducted in clinical studies to do what? To determine the optimal dosage, because we know that there is what? There's the minimal, and then there's the maximum dosage that can be used or should be used for the patient. So we just don't want to use the maximum, or we don't want to use the minimum. We want the best therapeutic outcome out of what quantity of drug we give to our patient. So as a result, we use this clinical trial to assess for dose optimization, because also in preclinical studies, as we will talk later, we will see that there is a time of dose optimization. This is one of uh, the early stages when somebody develops a drug. One of the early stages they will have to assess preclinically is how can their dose be optimized. And also in humans, just because it has been seen in animals, then it also has to be proven among humans that this is the correct dosage. So we have to check on other items like the frequency of administration. We also have to check on the route of administration, which is the better. Is it right to give this one orally? Is it right to give it subcutaneous injection? Is it right to give it through the buccal cavity, or whichever way that we see that this drug is able to be used maximally? Then the therapeutic window. We know that the therapeutic window is the time through which the drug is given to reach its effectiveness before it now falls back, it stops being effective in the human body. So we have to check for uh, and that's calculated by the effective dose over the little dose. So when we are assessing the therapeutic window, we have to assess what is how is this drug going to ensure that it easily works within the timing. Let's say this drug reaches its maximum concentration, which we usually use the AUC curve, the area under the curve, to assess the therapeutic window. So once we check that, we can know, okay, this drug, it is a fast acting drug. Maybe within 30 minutes, this drug will be already, it has already finished its work, and then it is out. So in that case, then we have to ensure that we keep the timing between the therapeutic effect and before it comes to a point of the toxic effect, and that is the therapeutic window. So this can be determined only when we use the clinical trial in humans. So the fourth point I'm going also to speak about briefly is on the regulatory approval. So we know that we do clinical trials to ensure that what the data generated from clinical trials is submitted to regulatory agencies for approval, and without successful clinical trial evidence, no drug can be marketed and physician cannot prescribe them. That's why we that are in this foundation, I thank the leadership that have made it very important and they have taken up the burden to ensure that among upcoming African students or upcoming African community, there is a desire for research, because it is only through research that we can break the barriers and the boundaries. So it is possible. There's a lot of, you know, a lot of things back home or in Africa that we can even use, and they can produce drugs. We have a lot of plants there, but we cannot say that, okay, this thing can be used as a plant because our fathers used it, now let us prescribe it. No, that could be used before, but currently it becomes very tough because of the very stringent regulatory approval. So as a result, it is important that we that are in this inclination of clinical research know all this about the regulatory approval. So also we go to another necessity for the clinical trial. It is what? It contributes to innovative therapies, personalized medicine, precision medicine, new treatment guidelines. So I think this is very clear to understand. So the clinical trials here, it has done what? Brings about innovative therapy. For example, I'm saying drug A is good um, in treating a condition, and then let's say we say pioglitazone, yeah, it's good, which is an anti-diabetic drug, it's good in treating diabetes, and then somebody comes and says that if you add pioglitazone and another saraglitazone or saliglitazone, it becomes a better drug. It has a better therapeutic outcome. Then, in that sense, this is an innovative therapy. This combination. Instead of taking one drug, we give the patient a double therapy. Then that means that the patient will have a better therapeutic outcome. So that is the innovativeness that we are trying to assess through uh, the clinical trials. You can compare 2 versus 3 drugs, 3 versus 4, 1 versus 2. So whichever way that you want to assess this, and then we see here in personalized medicine, and then it's kind of a very uh, upcoming concept. And then personalized medicine is usually that it's being proposed that for every individual it should be personalized. But then of course many bodies are still to approve this, because that means that every person, it's like they have to make their own kind of medicine based on their own um, kinetics and dynamics, but then on genetic basis, but that cannot happen. That's why it still hasn't been proven. But then the concept behind personalized approach is that every patient, you don't give the medicine based on that, on on just a general overview. You give the medicine after assessing the personal uh, the personal profile, the kinetics, the dynamics specifically of the patient. Then you go ahead to assess, okay, among these classes of drug, which one am I to give? When we talk of precision medicine, we are talking about patients being given medication based on their genomic status. What is their genome sequencing? Yeah, so we are trying to be precise. So it's not that one it fits all. No, like one drug it means that it is working for all. No, we want to ensure that okay, based on the genomic analysis of this patient, what is the best suited medication for them, and then this also helps in new treatment guidelines. And then we see also here that clinical trials are very important to improve patient care. This happens by giving access to advanced therapies. They bring about better monitoring, and then they bring about improved standards of treatment. And then finally, yes, we'll go to the phases of clinical trials, which is preclinical. And then we have the phase one, phase two, phase three, phase four. Basically, preclinical, which is phase zero, is lab-based research to tell if a treatment is useful and safe. And then we go for phase one. So from phase one is where we have clinical studies, clinical trial. So we have 10 to 80 participants, and then we want to assess the effect of treatment in the humans. And then in phase two, we want to assess the safety and the dosing. But in phase two, we have 100 to 300 participants, and then we also have trying to check what is the effective dose. And then in phase three, we have assessing of the safety and the efficacy, which is 300 to 3,000 participants, and then we confirm the benefit of this drug. Then finally, we have post-approval, which is what we call post-marketing surveillance as phase four. And all these five stages that are seen here is what takes uh, 15 years to 10 to 15 years to come out with a new medication or with a new drug. So uh, I've explained a bit. So here also we see that the main objective, we evaluate the safety, tolerability, pharmacokinetics, dynamics, the pharmacodynamics, how the drug, acts in the body, and pharmacokinetics, how the body is responding to the drug. So we see that it's very important that all these parameters are known, because if we don't assess all these things, then we are not being able to present the correct, precise medicine to the patient. So we see that the key features of phase one is that we want to do what? A dose escalation study and careful monitoring. In fact, you discover that many times the dosage given in preclinical very different from the one that we use in humans. So as a result of that, we want to assess, okay, if this dose is given, what if we reduce? What if we increase? What happens? Then there is a very strict monitoring in phase one, and that's why we have usually very few number of participants, usually 20. And then this is majorly what we call the pilot study. Pilot study is like when you are trying to prove it fast, when you're trying to prove that study first among humans. So it's called a pilot study, pilot clinical study. And then we are seeing here that it is usually conducted in specialized research centers, unlike phase three or phase four, which usually happen maybe at home. The participant can come and then they go back home, and then you assess them. In phase one, it is very different. They have to be there so that we ensure that the work or the medication they are given is working correctly, so that there won't be any issues. So monitoring is very important in this phase one. And then phase two, we see another concept of phase 2A and phase 2B, because we have said phase two, it is efficacy and then safety. Phase one, safety; phase two, efficacy. So in phase 2A, we have the early phase, and then this early phase is what we call the pilot clinical. Just like I've said, 20 to 200 patients. It is not multicentric, meaning it is done within one hospital only. Multicentric means different centers or different hospitals. So it is done single blind comparison with the standard drug. Single blind basically means that the person giving the medicine, who is usually the principal investigator, will be aware, but then the participant is the one that is blinded. The participant is the one that is told, okay, we are giving you drug and we will give you something that is dummy. So you take whatever we are giving you and then let's see. So the one giving the drug, which is the principal investigator, will know this patient is having this, and they will know this is the correct drug. But then they will also know which is the placebo. But then for the other one, the double blind, both of them are blinded, both the assessor and the the participant receiving the medication. A third party is the one that is able to come and know, okay, based on the randomization that is usually done in double blind, then they will be able to know that, okay, this certain number of participants have taken the therapy that we want to assess, and then this other one they have taken the standard drug. So we see in phase 2b it is done as a late phase. It's a pivotal clinical trial. It is 50 to 300 patients, and then it is multicentric. So we see that in summary, phase 2 clinical trial basically we want to assess what the efficacy and then the safety of, and then we go to okay, the key characteristics we see here is what it is randomized. Randomization is very important. Why? Because when you're doing a clinical study, you don't want to have bias. Bias will make your study very weak. You can't publish your papers, you can't present your papers in highly qualified conferences if you are not doing something as simple as randomization. Randomization is there to ensure that there is no bias. So the participant and the person or the principal investigator, they don't know which participant is associated into uh, group A, or which is the comparison group, or the standard care group that only do not receive the intervention. So in that case, it's usually done by AI, which also we will talk later, by a random application software. They will give you a sequence of numbers, and then you give the participant those numbers, and then you randomize them based on the number. So if you say, let's say from 1 to 10, there are 10 participants in your study. Number one, possibly the software will say number one will be intervention group, number two, intervention, number three, control group. Control group means standard care. They are not they are receiving placebo. Number four, intervention. So based on the sequence produced by the random allocation software, we go ahead with it. And then we go to phase 3. We see now this is where there's a huge population. It is between 1,000 to 3,000 participants. And then we see it's a large scale study to confirm the efficacy, because we said phase 2 we want to check the efficacy. Phase 3 we want to still now confirm that efficacy. And then the importance is that the results from phase 3 form the primary basis forward for regulatory approval, and then for drug labeling, clinical use recommendation. So from phase 3 is where now we can confirm that, okay, this medication now is to be used effectively. It has proven to be efficacious. And then um, yeah, we go to the next phase 4, which is usually post marketing surveillance, and then it is a long term safety monitoring. So here the participants have gone home, they have taken the drug. Now, for example, we say drug A came into market, it passed phase 1, phase 2, phase 3, it has been approved, people are using it freely. Then now we want to assess what is the long term effect of this drug. Does it have any drug drug interaction? Maybe there have been a consistent reporting of specific ADRs, and then they want to now assess this drug. Is it linked? Is the drug linked to be the causative agent of this? So that is why we have this post-marketing surveillance, and then it usually take a lot of time. And then it starts by recruiting participants. You monitor their drug use, you collect the data, you analyze the result, and then you report the findings. Maybe when a participant has used this drug for 10 years, this is how they start behaving. When a participant uses this other drug for a hypertensive drug for 5 years, that is how they start behaving. So that is why we have this phase 4 to solidify the reality or the long-term safety of these medications that are in use, and if they are having any adverse effects. So what are the challenges in conventional clinical trials? So now we are going to, of course, the as we are moving deeper to understand more about this clinical trial. So conventional, and then the other concept is artificial intelligence or modernized clinical trial. So a conventional clinical trial is kind of the most common one that has been used, where a participant, you have to bring them to the hospital. Okay, notice that this participant, they have whatever sickness that or illness they are going through. You measure their biomarkers, you measure their clinical parameters or biochemical parameters. Then after that, now you initiate your trial once they have given you the approval. So we see that the issue with this conventional clinical trials is what? They are of high cost. So drug development can do what? Can exceed US dollars 1 to 2 billion overall, especially in phase 3, because we said it carries the most number of participants, 1,000 to 3,000 participants. So we see that the major cost factors is what? Patient recruitment, investigator fees, site management, data management, monitoring, regulatory compliance, lab investigations, insurance. And then we see that due to this high cost, there's limited innovation, there's delayed development, increased drug prices, and then there's restriction for smaller companies. That's why we need what? We need AI, because AI can be able to lower the higher cost. Yeah. The secondly, we have what? Longer duration. So clinical trial often require many years, about preclinical stage 3 to 6 years, and then 6 to 8 years for clinical stage, and then regulatory 1 to 2. So we see that slow recruitment, regulatory approvals, data verification, long follow-up, and then complex protocols. All these things are some of what the challenges that are being faced in the conventional clinical trial. That's why today we are introducing the concept of what? AI in clinical trial, because we don't want to go through the systematic way of conventional trial, because it is a tedious process. One, we have seen it is having a high cost, and then we are now seeing that is what? Taking 10 to 15 years, as you can see in this photo that I have attached here. 10 to 15 years for the drug to be developed, and then of 90% of the drug entering clinical trials ultimately fail. 90%. So the success rate is very, very minimal for any drug that enters a clinical trial. So the consequence of this long duration is what? The more you are doing a clinical trial, the more money you are paying. The delayed patient access, because also there's something we call patient dropout. Many patients will feel tired, will be like, no, I don't want to be here. So we need AI to help us what? Overcome these long processes. Then, yeah, I've talked about the patient dropout or patient withdrawal. What are some of the reasons? It's adverse effects, lack of perceived benefit. You have told this patient, if you take this, for example, if you take maybe vitamin C plus vitamin D, if you, I mean vitamin C and E, if you take these supplements into your body, they are going to be helping you to have a better memory. Yeah. So the patient is having that hope that from time now, let me see, 1 month, 2 months, 3 months down the line, there is no change. They will feel tired, and then there's what? They will stop that clinical trial. They will stop going through that process, because there is no perceived benefit. When there's travel burden, when they have to be coming to the hospital, because you usually in a clinical trial you have something we call study time points. In study time points, you have to assess the parameters at inception of the study. Yeah, at the start of the study, you have to check. Okay, this patient, their blood pressure was this, and maybe at the second month, at the fourth month, and then at the end of the study, then you compare all this and assess what is the trend, and then uh, what is the outcome if you compare the final and then the time of inception into the study. So we see that these are some of the causes: loss of motivation, financial issues, all these things, they cause the patients to drop out. Then we see also there's what we call the recruitment failures. It is difficult to what? To enroll sufficient eligible participants. If you want to publish in all these very good journals, Nature, you need what? To have a study that has a lot of participants. And if that is the case, then you need what? To convince many people, because you're not going to do it on your own force. You are not forcing them. You have to convince them that this is the way. Please come into this trial. You will benefit from it. And it is difficult to do that. Why? Because there's what? You have an inclusion exclusion criteria. So you are saying these patients, maybe they have ever undergone surgery, they can't be included. If they are pregnant women, they can't be included. If they are lactating mothers, they can't be included. Children under the age of 18, they can't be included. Sometimes above 80, or what we call specialized population, they can't be included. So this inclusion exclusion criteria on its own, it has disqualified many patients based on what? Eligibility screening. Then there's lack of awareness. Patients can come and they don't know, in a hospital they don't know if there's any clinical trial. And then there's geographic barriers, distance, then there's fear. The patient is very afraid of their safety, of placebo use. What if any other thing happens? Then we see competition between trials. So for example, in where I'm doing my research work, I'm not the only PhD candidate doing a clinical trial. There are many others doing their clinical trials. So it is like you are competing for the same patient, because they are coming to the hospital and you need to meet them. Although of course it is in different specialties, but then apart from you, there's somebody else also doing a clinical trial in another case, and you can't bring a participant from another clinical trial into your trial, because that way the patient is already what? Is having bias. Yeah, they have already taken that medication. If you give them, the effect will not be produced. It won't be produced as expected. So we can't bring them into our clinical trial. So we see here there's recruitment failures. So now we go to the major thing, which is the artificial intelligence. So we are seeing that we have discussed so far the introduction to clinical trials, what are clinical trials, what are their phases, and then we have now spoken on the what? On the challenges that are being faced by what we call the conventional clinical trial system. So in that conventional system, now we are trying to beat that system by doing what? By bringing artificial intelligence. So this artificial intelligence, we see that as defined here, it refers to the ability of computer system to simulate human intelligence by performing tasks such as learning from data, problem solving, decision making, pattern recognition, and language. So we see basically that um, it's artificial intelligence. I believe all of us we have used it at some point. It is a broad field that enables a machine to do what? To mimic human intelligence. I think the very most common uh, one is ChatGPT, which anybody and everybody can use. You just ask anything, and from the available online sources, millions and millions of research papers of works that have been published, they are able to give you a concise answer based on what you've asked. So it is that ability that we want to use to overcome these stresses that we are facing in a clinical trial. We see also in machine learning we use, which is a subset of AI where systems learn pattern. So in machine learning is pattern what? Recognition. They follow a specific pattern, they notice it, and then from the data without explicit programming. So in machine learning there's what? No particular programming, but as a result of uh, specific pattern that is being formulated, then it now gives an outcome based on a specific data set. And then deep learning is um, sorry, when we are using the artificial neural network for complex data analysis. So it is going further much deeper to ensure that there is data analysis, that whatever is being given is not just any unverifiable or data that cannot be much repeated. So that is a very brief concept. I will not really talk much about that except for the artificial intelligence, and even for that I would really want to stick on AI in healthcare and specifically in clinical trial. So we'll go to the evolution of AI in healthcare. So we see here, if you look at the right side of my PPT, there is a very nice diagram there that I've placed on the evolution of AI in healthcare. So we see that first in the early computer systems, they came about in the 1970s. Then we have the rule-based system that came about 80s or the 90s. Then the 2010s, around the 2010 those region is when we have what deep learning breakthroughs, and then today those deep learning breakthroughs have now advanced, and by the help of artificial intelligence, they have become things like what we have smartwatches that help in healthcare assessment. So we see that in the 1980s, it is what expert systems that were used. For example, they assisted physicians in diagnosis and treatment recommendation. But then in the 2000s, the AI evolved from what? From fixed rules to data-driven learning models, and machine learning enabled prediction of using large clinical data sets. So this can be applied using disease prediction, which is very common right now. We have, for example, if you want to do a study on CHD, cardiovascular heart disease. So we or cardiac heart disease, we have to. They are very common disease prediction models. So we have the what? Sorry, I'm a bit forgetting. The most common one that we use is Framingham, the Framingham risk score. So these disease prediction models are the ones that based on the participant parameters. So, for example, my master's work was on the Framingham risk score. So this is a prediction model that was used in US. They used about 60,000 data sets to create a prediction model for how and who is likely to develop heart failure in the next 5 years, in the next 10 years, in the next 20 years, and above. So using this Framingham risk score, you are able to tell that this patient, based on their age, based on their gender, based on their blood pressure, based on their BMI, all these things, they are factored into what? Into their likelihood of developing a disease. So also we see they can be used in drug discovery and then patient risk assessment. So this is in the 2000s we have rise of machine learning. And then we see why is AI important in clinical trials. So we've been talking about majorly clinical trial, but why is this AI needed here? Why do we need it here? One, it reduces the trial cost. It accelerates the recruitment. It improves the protocol design. A protocol, just to talk on a little bit, a protocol is a document that you need to have, a systematic document that is going to be followed, the procedure that is going to be followed throughout your study. So let's say your study is going to be a 1 year study, then you need to have a very clear protocol. Okay, at month zero, what are you collecting? Maybe you say I'm collecting blood sample, 2 ml or 5 ml. Then when is the next time you're collecting blood? Apart from that, how are you going to collect? So the protocol, it assesses every dimension of your research work, and then it has to be written, because if it is not written, if it's not documented, it is assumed that it never happened. So we have to have a very clear protocol that has been designed well, and then we have to see that AI is important in enhancing patient safety. It supports personalized medicine. It also improves the data quality. And then so we go here for what AI technologies are being used. We've spoken on machine learning, deep learning, the natural language processing, and then we saw that the machine learning in clinical research is used particularly in supervised and supervised learning and supervised learning, when there's reinforcement learning, and then when there's what? Predictive modeling. And then there's used in risk prediction, there's outcome prediction, biomarker discovery. Yeah, for example, currently amyloid beta or p-tau proteins, they are very common. They are the common biomarkers that are used for Alzheimer's or neurological. Any neurological disorder, mostly they will ask, okay, is there any amyloid accumulation or amyloid beta deposition there? Is there any p-tau? Now, we are seeing that through this machine learning, newer biomarkers are uh, be, being being discovered, and they are being introduced into the market. Even the technologies themselves, such as the Seymour technology, these are helpful to ensure that it is easy, and then it is very to the point. It is very specific. Yeah, that is also very important, especially when we are dealing with risk prediction. We assess the specificity and the sensitivity so that to ensure our work is not overstretched or our outcome is not overinterpreted. Then we have the deep learning, the neural networks, CNN, RNN, and then transformers used in medical imaging, ECG interpretation, genomic analysis. So they usually use deep learning, and then we have the natural language processing, which we see that it happens by what the extraction of health, uh, electronic health records data, and then clinical note analysis, trial matching, adverse event extraction. So generative AI. So now we are going a bit more on this AI in clinical trials. So we see on this photo that I've given here, we see that uh, how this progression is expected to grow from 2023. I mean, from 2023, that's right, to 2033. And then we see that the forecasted market size for 2033 is what is US dollars 140 billion for what? Generative AI. Generative AI is where you just put your data set, or you ask any question and then you receive whatever you want. So the machine itself should be able to produce or generate the data based on what you have given it. So, for example, we are seeing here it can work on protocol drafting, concept form generation, when we are seeing for the regulatory documentation, also trial simulation, and then we also now see that large language model based systems, they are also increasingly used in what? Concept document drafting and protocol automation. So this is about generative AI in clinical trial, and we will continue to talk more on this. Then we see now digital twins and in silico. So now we are talking specifically on what AI in the clinical trials and how it is currently being used, because as the days are being progressing, newer technologies are being used. So the other thing, or the other way that AI is being used in clinical trials, what is a digital twin. So a digital twin is, imagine you have a health record. Health record, electronic health record, is basically when the patient comes, usually the data that they give is usually, it remains within the hospital. So that electronic health record is there, it's kept, and by different hospitals have different timelines. Some may be within 5 years, within 10 years, they are able to dispose all that data. But then there's a specific timing that all those um, patient records have to remain within the hospital. So we see that a digital twin is they use that health record to create a model similar. Let's say a patient has the patient is having hypertension, and then the patient is having maybe uh, 140 over 90 as the blood pressure now, or 160 or 180, and then it is kind of growing higher. So all that patient profiling, the age, the gender, the body mass index, all those factors, the genomic analysis, then they are being brought as what? They are brought into a model. A model is formed to act as that patient. So that becomes a digital twin. So that it will, instead of you using a patient, a physical patient, you use a model. You use a specific model that will act as the patient, and it will give you the same outcome which would have been seen in that patient, because it is the same parameter that they are having. So that is the same thing that we are having. What it's called, what? A virtual patient model. So it's not a real patient, but it's a virtual patient, and then it works on computational simulation. So a simulation is, you are trying to mimic a patient, but it is not a real patient. And then we are saying it can be used as what? Synthetic control arm. So especially when we are having uh, these large clinical trials, and maybe you don't have many participants to put in the control arm, because we have the intervention group and control. In the intervention, you are giving them the drug or the intervention you want to see, but then in the control arm, you are trying to give them the continual or the standard therapy. Now, if you are having, let's say, for example, fewer sample size, you can ethically go for this synthetic control arm because they are not receiving any intervention. So the outcome will be similar to uh, the other participants, or to the twin or the real patient that was to be used in that study. So uh, I'll go to the uh, next one, which is the AI in clinical trial design. So we have talking we've talked about the digital twin, and then now we speak on clinical trial design. So when you are making any trial, any clinical trial, you have to make a very robust trial design, because based on your trial design only will you be able to get the required efficacy. So we see that it uses what? Adaptive trial design, which is different from the conventional trial design. Our conventional trial design is, if you have decided it is A, or it is step one will be followed by step two, by step three, by step four, then you are not permitted to break the protocol. In fact, if you do that and you submit your paper for publication or your thesis, the ethical committee can reject that work and legal action can be taken. Why? Because the protocol has been broken. You have gone against what was expected of you. But then when you use AI, we see that there's what? You do a background review, there's adaptive trial design where you clearly state what are your objectives, what your primary outcomes, what are your pre-planned adjustments. So in your adaptive trial, you can change. You can change. If you see in that in this photo I've attached here, when there's in the enrollment phase, there's patient accrual, and then we see updated variation parameters. So you can update. Okay, you see that maybe one of the biomarkers that you have used in your model is not giving any outcome. It's not, it is a futile effort. Then you change and put another model, and you're still trying to do that kind of trial design. This you can't do on a normal human being, but in the model you can repeat, you can do trial and error until you find. Then you see after updating it, we go for the interim analysis. If the interim analysis is not to be done, then you go for what? Final assessment. If you have to do it again, you go for early stopping, which is as a result of what? Of the efficacy and the sorry. So that is with regards to clinical trial design. So basically, the major difference is what? In conventional clinical trial, there is no adaptation, but except in very rare circumstances, and then you still have to meet the ethical committee and write to them. Otherwise, in the clinical trial design by AI, you can do adaptive trial design. So we go for the protocol optimization. I've already explained to us what a protocol is, and then based on this slide or this photo that I've attached here, you can see that 60% of protocols require what? Amendment. When you are even submitting for a research grant, yeah, your protocol is what convinces people. What you have expressed through that protocol. This is how I'm going to do A, B, C, D. The clearer your protocol is, the better. And now you see that it's not everybody, or sometimes people are not able to really go deeper. And as humans, we can miss one or two things. And that's why you see here 60% of protocols, what? They require amendments. And that on itself is what? It is costly. It is taking time, because to make a protocol from scratch is not an easy thing. To amend it again, to present it to the ethics committee, it's already taking a lot of time. So you can even see from protocol design to patient recruitment itself, it's taking the traditional clinical trial route is taking almost 8 to 12 years, and this is slow and inefficient. But when you use AI, we see that what? there's 85 prediction accuracy. You just give the AI the tool, the tools, whatever you are wanting, or whatever your hypothesis is, whatever your objective is, what is your title, and then what is the expected outcome, and then based on that you specify your inclusion exclusion criteria. They can give you a protocol that has been designed with what, with 85% prediction accuracy. The same thing happens with what, with patient recruitment based on the um, healthcare healthcare records that are available. The AI can direct you based on it. These are the patients that are in this hospital. These are the patients that are having hypertension stage 1. They are having hypertension stage 4. And if your study, you're going to be measuring on hypertension stage 4 patients, then you don't need to um, waste your time seeking maybe other patients. You can call them and then enroll them. And this what reduces the time of patient recruitment. So it enhances it faster by 3 times. That's for protocol. Now we see for AI for site selection. So site selection is also very important when you are dealing with um, with a clinical trial. Why? Because if your site is not correct, if your site is not able to produce the required number of cases, one, if your site does not have the maximum or the standard equipment, you can't say you want to run a clinical trial on neurology patients and your hospital is only it only has cardiology department. That does not work, or it is only having a maybe another kind of specialization there. Maybe it's only for pediatrics. You can't do that. So the site has to be selected uh, carefully to ensure that once the patient recruitment start, the infrastructure is there, the demographics are there, then the people that are supposed to do it, they are what? They are available, because it's not just important that you have the facility. You have to ensure that the people that are working on this clinical trial, are they really standard to it? And then there are many different types of AI selection tools that are being used, and we will also just talk on them briefly as examples. So we also go for the predictive modeling, and in this predictive modeling, we see that AI has been used to predict the recruitment success, and then it also has been used to do what? To predict the dropout rate. Also, it's been used to what? To predict the adverse event probability, and also it has been used to what? To assess the trial completion likelihood. So this is predictive modeling. Just like I earlier on informed us that um, when you use AI, you can use it to predict this patient will have maybe heart failure or something of that sort. So the same way, we through predictive modeling, we can try to assess, okay, will this patient even finish this our study, or will they maybe stop midway. Now we'll speak on also the synthetic control arms, which I've already also spoken a bit, and then the synthetic control arms, which is um, what we spoke about, the digital twins. Yeah. So it uses what? Historical data, and this historical data is what? Real world evidence, because this historical data is whatever that has been kept what? In the electronic healthcare records, which are kept usually in the department, in the hospital. And then this helps to reduce the placebo groups, and then we see that it does what? The advantage is that there's what? Ethical improvement, and then there's faster recruitment. You don't have to wait for more and more participants to give a yes, because you already have dummy, or you have models that can act as patients, but they are not real participants or real patients. So also we see that AI can be used as what? In adoptive clinical trial. So we see that in this utilization, it does what? It enables dynamic dose adjustments. So we were talking about precision medicine and then personalized medicine, and we see that dynamic dose adjustment is one of the key parameters that are being assessed also, and they try to overcome it. When you as a physician or as a clinical researcher, you want your patient to have the best therapeutic outcome, then we have population enrichment, real-time analytics. So we see that from the group sequential analytics through the adoptive clinical trial. We can also have the adoptive enrichment. We can have the sample size re-estimation, because we have said, unlike uh, the former or the old way of doing conventional clinical trials, in adoptive you can change your sample size. Before it is 100, you write your research paper and they say 100 participants we don't accept. You can increase your sample size, you can change and adjust your sample size even in the middle of the study. If you discover that the power of the study is not going to work out, if you discover that maybe this the population base is not working correctly, then you can change few things, because you have models and not humans per se to use. So that is how AI is very important in adaptive clinical trial. Now we also go to AI in patient recruitment, which is very, very important. And we have talked about this patient recruitment being one of the things that takes a lot of time. And we see that 85% of health systems, they leverage on one AI. So it is something that is becoming accepted more and more and more as time goes by. So we also need to be up to date on what these things are and how we can benefit from them. So we see that the AI, the artificial intelligence, will do what? It will scan the um, electronic health records, it will scan the registries, it will scan the claims data, it will scan the what? Genomic databases, because all this, especially in equipped, uh, very equipped and large hospitals, all these data sets will be available. Data is very, very important. We'll also talk about it as we continue before we finish. The healthcare data or the patient data is not something that just is there to be discarded anyhow. No, it has to be well taken care of. And yes, also AI is used, yeah, just on this, also in patient recruitment. So we have, especially like for those of us here in India, we know the IQ VIA, the group itself. It has its own modeling, or the Deep Six AI. These are what models that have been used to do what? To enhance this patient recruitment, and they are very trial X, some of them, and they are very, 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 very much important currently, that are being used even over here in SRM, being used to bring participants. So it is necessary we learn about this. For the NLP, natural language program based eligibility matching, we see that this works by what? Extracting eligibility criteria. We know um, in a clinical trial we have what? Inclusion, exclusion criteria. So based on the exclusion criteria provided, they will do an eligibility marking. Based on the inclusion criteria, there is an eligibility matching, and then that way you can be easily matched with the participants you want, and then that does what? Ultimately improve the recruitment speed. And then we see that there's what? Diversity. That's one of the things, even for the Framingham risk score, which I've talked about as one of the tools that uses predictive modeling. Yeah, it was only used in the whites or in the Europeans. So you can't use the same model and expect the same outcome when you use it among Asians, among Africans. No, it doesn't work like that. That's why it needs to be re-stratified. That's what happens. So that data is added onto other data sets from these underrepresented populations, so that they can help do what? they can help make that model to be more robust and ensure that it works out correctly. So we see that through AI, we are able to what? To assess the patient, because even when you are bringing the patient into any study or patient is being admitted, their staff, they will ask like what is their ethnicity, their color, and all these things. They will be used for what? For diversity and inclusion. So you don't have to go through maybe 1,000 health records to assess, okay, this patient, which ethnicity were they? Were they European? Were they Asian? No. Through AI itself, you are able to do what? To identify the underrepresented population, and then you are able to match and know these are the ones that are available, that are needed to be included in your study. And then moving on, we go for the remote and decentralized clinical trials. So through AI, we have AI powered. We can have what? Telemedicine. Telemedicine is one of also the really upcoming fields, I should say, particularly in healthcare. So telemedicine is where you don't need to see a physician, because conventional clinical trials, the patient has to go to hospital, meet the clinician face to face, and but right now there's remote monitoring. It's just you either take a call, or through a wearable sensor, or through a mobile app, you send that data to the hospital, to the clinician, and then the clinician at the same time they will send the report back to you on what to do. Or let's say you're having an ADR in a clinical trial. So instead of you going to meet the clinician, you just call them telemedicine. They will tell you stop maybe the medication or take it at this time. So all that is what we call what? Decentralized. So instead of having many participants coming to one place, which is the hospital, for them to have the clinical trial, we have through AI been able to what? To decentralize. So that helps to what? To reduce the cost, the timing, and also it enables the patients to be in their comfort as they participate in these clinical trials. So we see also we have AI in data collection and monitoring. So example, we know the smartwatch, the ECG patches, we have the glucose monitors, all these they are wearables and they are digital biomarkers that are used for what? They are used to collect data. Nowadays, even Samsung, you just wear your watch. Your watch can tell you your BP, can tell you what is the risk of you having heart failure, what is LDL, HDL level, all these things. They are really helpful, and it helps you to ensure that you keep a healthy lifestyle. So the AI can be used in what? Data collection and also monitoring. Glucose monitors, it monitors you are having a diabetic patient, they need to be doing time to time assessment. So with these AI tools, we are able to ensure that the patient's health is being monitored clearly and very, very systematically. So also we see real-time monitoring, which ensures that there's automated alerts there is monitoring. So let's say for all these wearable sensors, participants are able to, let's say you have included them in your clinical trial, they receive alerts in their phone, they receive alert in their wearable sensors, and then they are able to know, oh, I need to be taking my medicine right now. I need to maybe start running. That watch is showing that there has not been, activity. So the patient has to do this stuff. So AI is also very important in real-time monitoring. And then it's also very important in risk-based monitoring. It assesses, is there any protocol deviation? You said in this study, the participant should be sleeping for 8 hours. But this participant you have included in your study, they are only sleeping 4 hours or 3 hours. It will show through what? Through the wearable sensor, or through the monitoring that is being done, or through their biomarker analysis. So all this is there to ensure that the risks that the patients are being exposed to, you can monitor them. And if there are any inconsistencies in the data, you can also pick it out. You can pick it out. You don't have to wait until they come back. No, just at the time there's an inconsistency in their data, you are able to pick it out. And then we also see that there is AI is now being used very much in imaging trials, in oncology imaging, in neurology, in radiology, and in neurology, cardiology, all these things.
So we see that imaging has really benefited as a result of what having AI embedded, particularly in clinical trials. Then we also see that AI has been used for advanced event detection. So usually we have what we call uh, the WHO monitoring center, which is in Sweden. But then through these AI tools, we don't have to just wait until a patient calls and no tells you this. No, through the patient reports, that is one of the ways. But then the AI can extract adverse event just based on the health record, and then from the physician note. So physician is writing, and then they say this patient was experiencing this and this and that, and the physician has not even himself or herself understood what it is. But then the AI itself is able to pick out that adverse event, and then you assess, is this patient really supposed to be on this study? Because also in clinical trial we have to assess for adverse events, and then if the patient is severely affected, then they have to be stopped from continuing in that clinical study. And then we also see that AI is used in big data analytics, such as genomics. Also, we have spoken about emergency health records, the wearables, the claims, and then we have what we call the predictive analytics, and then the AI for statistic analytics. So predictive analytics is where we are doing what? We are trying to assess the progression. This patient has this cancer tumor cells. At After 5 years, how is it going to progress? After 10 years, how is it going to progress? What is their response rate to this medication? What is their response rate to chemotherapy? What is their response rate to maybe immunotherapy? And then what is the survival outcome based on AI, based on the data set that has been fixed in that model? It can give you the idea of what is the chance of survival of the patient. And then we also see that AI can be used for statistical analysis, especially when you are handling missing data, or when you're trying to do a pattern recognition, or when you're trying to do what we call complex modeling. Yes, and also this is very important. The real world evidence, AI, we see it integrates real world data, and then post marketing surveillance, and then the longitudinal outcomes. So we see that longitudinal outcomes is what? These are outcomes that are assessed over a kind of longer period of time, 2 years, 3 years, 5 years, 10 years. And then we see that the FDA increasingly emphasizes AI and real world evidence in regulatory evaluation. So when you are evaluating, or one, they are proving a, an AI tool, how good it is working, they have to assess what? It is longitudinal outcome, and this has to be what? From the real world evidence. And then we see federated learning, which includes multi-center data sharing and privacy preserving AI and collaborative learning. Then we'll go to the next chapter, I believe, which is almost towards the end, the regulatory and the ethical considerations. So in clinical trials, AI in clinical trials. So who are the people that are there to regulate it? So we have the organizations such as the US Food and Drug Administration, the European medical agencies, we have the WHO, we have the International Council of Humanization, and then the FDA guidance on AI is what? It released a guidance that supports regulatory decisions in drug development based on AI. So it approves it. But then the key principle is what? There has to be transparency, there has to be validation, there has to be a risk-based approach, and then we have to have life cycle monitoring. And then for EMA and FDA joint is that it has to be what? The AI has to be human-centric, the data has to be governed well, then model has to be validated, and then also what is the life cycle management of that particular AI tool. And then the ethical challenges that we face using AI in clinical trials is what? There's possibility of bias, there's explainability. Black box AI is when there's a lot of data that you cannot really assess based on what outcome this model has given you this result. So in that case, you cannot really change it and give the correct. The model is able to manipulate that and give the wrong output. The patient privacy also we see is not there, and then the informed consent, there's no place where they sign. Then the data privacy, which something I spoke about, the HIPAA, and then the GDPR. So this is for the US and for Europe. So these are the organizations that provide regulations for the data privacy and security, particularly with regards to health. So it is very important that AI tools comply with this, because AI is derived from having data and more data. And then we can see that there can be bias in AI models, such as poor data set, underrepresentation, and then historical bias. And then the consequence is what? There's an equal outcome, or the wrong outcome that is to be expected. So, for example, case study, AI was being used in COVID-19 for the vaccine trial, for the recruitment, and then for assessing for the safety. Currently, it's being used by many pharmaceutical companies, such as Pfizer, for what? Predictive analytics, trial optimization. So these are some of the current scenarios, real world scenarios of how AI is being used. So AI also, there are many startups that are being used in clinical trials, such as Medidata, Deep 6 AI, Sama, Tempus, and then in silico medicine. Then we also have AI in oncology trials. So these are what they are based on: biomarker one, imaging, and then precision oncology, which is kind of the best way to come out with very precise understanding of the pathophysiology, or the undergoing work in that patient. So when we can use these AI tools, we can be able to now target our medication. We can be able to target our therapy. So the future trend is what? Autonomous trials, agentic AI, digital twins, which we have said is going to be increasing, AI co-pilots, and then quantum AI. And then what are some of the challenges and future direction? The current limitation is what? Lack of standardized data sets. So the data set, usually it's very difficult to have standardized. So the ones available is the ones people use. So that is a limitation. There's regulatory uncertainty. So you can work on a model and then it gets to be rejected because of something. So it is uncertain about it to be accepted. And then we have high implementation cost, limited explainability, what I spoke on, black box AI. And then we have AI regulation, which remains a major challenge due to validation and accountability concerns. So the future of AI in clinical trials is predicted as what? There will be fast approvals, there will be more decentralized trials, there will be reduced costs, there will be personalized medicine expansion, and then AI may significantly reduce development costs and improve trial efficiency. So my conclusion is that what? The AI impact on clinical trials is what? There's a 65% increment in patient enrollment, there's 85% accuracy in predicting trial outcomes, there's a 40% reduction in operational cost, and then there's a 50% faster trial timelines. So we see that AI is doing what? It's transforming every stage of a clinical trial. It has improved efficiency and patient centricity. So the ethical and the regulatory oversight is still remaining very essential. And then we see that in future, we are predicting, or we are looking to a scenario where AI or clinical trials will be very much using or including AI in their working. So thank you so much to everyone that has been listening to my long presentation, and most especially thank you to the team for giving me this chance. And yes, over to you, modulator.
Thank you. Thank you. Thank you, Mr. Ian, for that elaborative session. For sure, it was very productive. I want to thank people who are streaming live on our Facebook account and YouTube account. We don't take that for granted. And there is a question someone is asking from our who is streaming on the side of YouTube, and he's saying, he's asking that Beyond your scientific achievements, you have demonstrated leadership through mentorship and student coordination. What advice would you give to young African researchers and pharmacy students who aspire to pursue impactful careers in clinical research and innovation? Then another one is your research spans cardiovascular risk assessment, pharmacovigilance, and clinical interventions in chronic disease. Which area of pharmaceutical research do you believe is currently most underexplored but has the greatest potential to improve global health care? So those are the questions that are coming from the YouTube side. Then we have one from our Facebook. One from our Facebook is from Mr. Jordan. Yeah. You developed ScanRx and AI driven innovation aimed at reducing prescription errors through handwritten recognition and autocorrection. How do you see artificial intelligence transforming pharmacy practice and patient safety in the next decade? Yeah. Okay, those are the questions. So I don't know if you've got them or I can say them again.
Maybe the first one only I didn't get them.
Okay. Beyond your scientific achievements, you have demonstrated leadership through mentorship and student coordination. What advice would you give to young African researchers and pharmacy students who aspire to pursue impactful careers in clinical research and innovation? That was the first one.
Okay, so if I have to answer the first one, I think thank you so much for the person that asked that. I would really recommend that you have a burning desire for research. Research first, you really need to have that desire in you, because to be very honest with you, there are many failures in research than success. So that means that you are likely to encounter more failures in your research work than more success. Ask any PhD student or any person that has done research even to masters, and they are even doing it. So the problem that I usually see with many young people that are starting their research is they do the first step, it fails, I'm tired, I'm discouraged. They write their first paper, I'm tired, I'm done, I'm not writing again. So they give up. So research is not just about the joy of doing this thing and passing. No, passing is good, but please have it in mind that you will face challenges. So be prepared early enough, and then be resilient that no matter what happens, I'm going to do something. I want to impact my world by my research. So as a young researcher, I would advise you to do what? To have it in mind that it's not just going to be rosy. So when you face challenges, be prepared for it. And also, whatever you think, try it out. Don't close your mind. I don't know how to put it. Try as much as research is about exploring. Explore, have a lot of knowledge. Think wide, think big, and then you will have many thoughts that will be springing up in your mind, and you have a lot of research questions. Yeah, I think that's what I'll answer with regards to that. Secondly, I think the individual asked about the most unexplored area. So in my master's, I did a work on cardiovascular diseases, pharmacovigilance. Right now, I've shifted. I'm doing my PhD work on neurology. So my PhD work is a clinical trial among neuro patients. So if I have to say what is one unexplored region, I may not be able to really say, but if I can mention one that you see also research, how can I put it now? Research, particularly as you keep moving higher, you discover that there's a lot of demand for research and for good research. And then in that essence, one of the areas of research in healthcare that has a lot of burden, that receives a lot of money, a lot of grants, because also if you want to delve into research, especially as you want to keep increasing your knowledge and widen your research experience, know that research is money, a lot of money. So you must also factor that. So one of the areas that is being explored and it's like an ending is cancer. Maybe that's one of the things I might say, because I see a lot of call for grants for cancer trials, cancer this, cancer that, in vivo, in silico. So cancer is like one of the really up there funding areas of research. Don't know if I've answered very clearly, but I've tried to explain. Then the third question will be on AI in pharmacy practice in the next decade. So my estimation is that in the next decade, especially if you're in pharmacy practice, I think it is very good we start preparing for learning models, and we also, if possible, learn how to build these models, and if not, then be very updated. Because, for example, now this scan RX that I presented, I was able to present it, but I've really not pushed far with it, and I'm still intending to push far with it, because right now I'm still working on my research work here. But then that's still to let us know that what I'm aware that in the next few years, even in kind of the whole hospital, things will be changing in the nursing. Of course, the healthcare professionals will not leave, but then for you to be at work there, for you to be on the safer side, you need to be very skilled. So gain as much experience on these on these AI tools in pharmacy practice that we can use as a pharmacist, compare medicine, look alike, similar sounding names of drugs. Such kind of tools, they are there. So be exposed. This also helps you in your CV, and it shows that you are a person that is interested in what you're doing. So that would be my answer to the third question. I don't know if I've really tackled as expected, but that's what I'll have to say about it. Over to you.
Okay, thank you very much. Now, those were the questions from the people streaming on Facebook and YouTube. Now, let's turn to those that are to the call on the Google Meet. Anyone with a question, raise your arm up if you have a question. Anyone with a question? Yes, Mr. Emmanuel.
All right, thank you for the well-moderated webinar for today. Mine, it's not a question. Mine, it's a humble appreciation to our presenter, Dr. Ian, for the well-elaborated presentation and thoughts as per the current clinical trial and guidelines. It's a journey that we have appreciated, and we look forward to have more fruitful engagement with you in future. And to the participants, definitely it has been well observed journey. To me, I don't have any question to ask you, but a token of appreciation from the sincere bottom of my heart. I thank you.
Thank you, Mr. Emmanuel, for that nutritious submission. Any other with a submission or a question? Then I want to pass this. Madam Ampride Praise, please, you will stay on the call. there is a communication that one of the directors wants to say to you. So please stay on call. Any other question? So if there is no any other question, I request Mr. Ronald to give a vote of thanks to our presenter. Then after Mr. Ronald's vote of thanks, Mr. Sembo Rogers will give us a prayer as we close off today's webinar. Mr. Ronald.
Well, good evening to you, ladies and gentlemen. I want to thank our members for having logged into this call. It has been such an informative session from Dr. Ian. We are proud to have you amidst us, and that was really a very great submission. So, to our members, hope we've all learned, and we see how AI is revolutionizing the healthcare system. With time, as we've been told, one drug would take years and years, 20 years to come up with one molecule. The tablet you see in market, the one paracetamol. This era of AI, or in this generation, where machines are being trained, to act like human beings or to imitate human beings, you can see that the healthcare system is being revolutionized. Now, much time that is being spent, as the speaker said, in drug discovery, clinical trials, it is all going to reduce. And besides clinical trials as well, there is a very growing trend of application of AI in the initial stages of clinical trials. The likes of drug development, lead identification, lead optimization, so on and so forth. I published one paper where we were using AI in drug discovery. It was about cardiology, myocardial infarction, whereby we were using data, feed them into different AI tools. AI, it has been with us for a long time, since 2010. That's when we started recognizing it. But all the way in the 2000s, actually, early 2000s, we are still living with AI, only that it has gained public recognition of recent. But otherwise, we want to thank you, and this is very important to all of us. We need to level up now the standards of the world. We can see the standards of the world, where the world is heading. Some say that AI will replace human beings, AI will replace doctors, AI will replace pharmacists, but it shouldn't be the case. If that is to happen, that will happen only if we've not leveled up ourselves to the standards. But otherwise, AI is coming as our co-worker, our side worker, whereby we shall be leveraging AI, because AI is, as it says, it is intelligent, but it can't match the intelligence of a human being. It is you to instruct this AI. It is you to give it instructions. So it is us who will be giving instructions rather than it giving us instructions or requesting us. And Mr. Dr. Ian, thank you very much for the submission. We hope to have you more to enlighten the community in research and research ethics and so on and so forth, so that the young generation can as well level up. The rest of the world is growing faster, probably, than some of our countries. it is all based on science and research. So with those few comments, I want to once again thank Dr. Ian for having delivered to us such an informative session, and to all our listeners, keep tuned in. More is still coming. A lot of knowledge is out there, ready to be dispatched to all of us. Thank you very much. Back to you, Mr. Pius.
Thank you, Mr. Ronald, for that very elaborated submission. And my word to Ian is, please, we as PASH is your home. If you feel like being with us at any time, the doors will be open for you. Please bring your expertise, as we are saying that let's build our home countries through the webinars, through the expertise that we get from outside. Have to share it with our people at home. So the door is open. Wherever you feel you should share your expertise or your research, the doors will ever be open for you. So next on the agenda, we have a closing prayer from Sembo Rogers. Please, Sembo Rogers, may you give us a prayer. And Madam, please remain on the call after others have left. Thank you.
Okay, thank you for giving me this opportunity to evoke the Almighty into this. So I request all of us to humble ourselves for a prayer. Almighty, we thank you for this opportunity that you have allowed most of us to access this information, to get the teaching, to get the research information from Ian. We thank you for all that will proceed, because you will bless it so that we can benefit from it. We request for your support. We request for all that is needed to go on smoothly as planned. So thank you, Lord. I praise this, believing and trusting in the name Almighty. Amen. I request the moderator to take over.
A very good evening to us all. Am I audible?
Good evening. Yes.
Yeah, I would request to stay on call with Mr. Ronald Sechkubo, plus Mr. Pass, the CEO, plus even Madame Praise. Rogers, we shall engage you in another meeting very soon. Thank you so much. May God bless you. So to my fellow leaders who are present today, a very good evening to us all. I hope I'm loud and clear. Mr. Ronald, I hope I'm loud and clear. Then Mr. Pius, I hope I'm very loud and clear.
Yes.
Pius, am I audible?
Yes.
Ronald, am I audible?
You're audible, sir.
Oh, okay. I congratulate your department for steering the great work